Hello, my name is Giovanni Camarata, and I was part of the GGC BioBridge course this semester. Today, I will be sharing a comparative analysis of various assays for evaluating antioxidant capacity and cell cytotoxicity of yerba mate. The purpose of this study is to gain a better understanding of the assays used to test antioxidant activity and cell cytotoxicity. These are important because reactive oxygen species, which I will go into more details on in the next slide, can severely harm and even kill cells in the body, leading to various health issues. Through a comparison of these assays, the goal is to determine the best assays to use for analyzing yerba mate. Reactive oxygen species are a type of unstable molecule containing oxygen. These molecules contain a free radical, meaning they have an unpaired electron, and this makes them highly reactive and harmful to cells. As you can see on the diagram to the right, the free radical is missing one electron and will try to steal from stable molecules to complete its octet. This can be very harmful because stable molecules will become unstable and our bodies like stability. This is where antioxidants come in. Antioxidants are substances that inhibit oxidation and counteract reactive oxygen species. As shown in the diagram, antioxidants will share a spare electron with free radicals to deactivate them and make them stable. This is why the study of antioxidants is important, because effective antioxidants can help our bodies tremendously. Here is some background of yerba mate and its significance to me. The scientific name of yerba mate is Ilex paraguayensis. It is a plant native to South America, and namely Paraguay, where it was first consumed by the indigenous Guarani. Yerba mate is consumed as a tea, either cold or hot, and is drunk mainly in Paraguay, Uruguay, Argentina, and southern Brazil. It is also known to contain various compounds which are known antioxidants, including 11 different polyphenols. Its significance to me is that I am Uruguayan, and mate is a big part of our culture. Mate is a drink that is consumed consistently all over the country. My family and I here drink mate a lot, and it is a staple at family gatherings. To the right is a picture of the Uruguayan flag and a picture of my father and me drinking mate. I will first go over two antioxidant capacity assays. These assays are done to examine whether or not various supplements exhibit antioxidant activity. The first assay we will look into is the Trolox equivalent antioxidant capacity assay, or TEAC assay. This assay uses Trolox, a water-soluble analog of vitamin E, and a well-known oxidant, as a standard to compare to a chosen supplement. The chosen supplement and Trolox are tasked with reducing an ABTS free radical. This first figure shows how the ABTS free radical is formed from regular ABTS into the free radical form. Good oxidants will reduce the ABTS free radical and cause a color change from bluish green to clear. This color change can be seen below, with the bluer solutions having a higher concentration of the ABTS free radical and the clearer solutions having a lower concentration. Since this assay is colorimetric, meaning a color change is involved, the results can be analyzed through absorbances from a spectrophotometer. Let's take a look at an example of some mock data for the TEAC assay. The data from this slide came from a TEAC assay done to compare the antioxidant activity of a strawberry with Trolox and quercetin, another well-known antioxidant. The supplements are tested in various dilutions, and their absorbances after reacting with the ABTS free radical are put into a percent inhibition. The percent inhibition is the amount of the ABTS free radical that has been inhibited by an antioxidant. A higher percent inhibition means a more effective antioxidant. The percent inhibition is calculated by taking the absorbance of the ABTS free radical remaining after reacting with the supplement and dividing it by the absorbance of the stock ABTS free radical solution, and then multiplying that by 100. In the scatter plot above, the y-axis represents percent inhibition, and the x-axis represents the percent dilution of various supplements. Since a higher percent inhibition means a better antioxidant, it can be seen that quercetin actually has the best antioxidant at each dilution, better than strawberry and Trolox. The strawberry was only better than Trolox. This means that strawberries exhibit antioxidant activity. Aside from graphically, this ranking can be seen mathematically by taking the ratio of the slopes of the lines of best fit for each supplement. Since Trolox is a standard, all slopes will be divided by the slope of Trolox, and it can be seen once more that quercetin has the highest percent inhibition, and strawberry falls in between Trolox and quercetin. One downside to this assay is that it doesn't reveal which compounds within each supplement exhibit the antioxidant activity. It's solely a comparison of the whole supplement. One upside to this assay is that it is fairly quick and easy to perform. Another assay is the ferric reducing antioxidant power assay, or FRAP assay. This assay measures antioxidant potential through the reduction of ferric iron to ferrous iron. When supplements react with ferric iron, a high production of ferrous iron means a good antioxidant. This assay is also a colorimetric assay in that the solution colors will change from clear to navy blue as ferric iron is reduced to ferrous iron. The absorbances of these reductions are compared to a standard curve based on the stock solution to give results in micromoles of ferrous iron. In the chart to the right, it can be seen that the green tea produced more ferrous iron than strawberry, which produced more ferrous iron than grape juice. This means that green tea was a better antioxidant out of the three supplements. One downside to this assay is that the result is just a molarity with nothing to compare to. This assay would be effective if you test a well-known supplement alongside your chosen supplement. Now I will review 4 cell viability and cytotoxicity assays. These assays are performed because it is important to know how the chosen antioxidant affects the cells and how toxic the supplement is to cell health. Here we have the colony-forming unit assay and the WST1 assay. The colony-forming units assay tests for cell viability. This assay is done by simply counting cell colonies to determine the percentage of live cells left after exposing them to the supplement. As seen on the right, the colonies are shown, and the top row is the control that was not exposed to the supplement, and the bottom row is the set that was exposed to the supplement. The cell viability is calculated by taking the number of cells from the experimental group and dividing that by the number of cells in the control and multiplying that by 100. The WST1 assay tests for cell cytotoxicity. This assay is done by adding WST1 to cells after exposing them to the chosen supplement. In the presence of active cells, WST1 will form into formazan, as seen on the reaction to the right. This is a colorimetric assay because there is a color change from pale yellow to dark yellow. The more formazan that is produced, the better, because that correlates to more living cells and an antioxidant that is healthy for your cells. Another cell cytotoxicity assay is the dye exclusion test using methylene blue, a synthetic dye. In this assay, the methylene blue is added to cells that have been exposed to the antioxidant. The principle of this assay is that live cells will take in the dye, will not take in the dye, whereas dead cells will take in the dye. This is a colorimetric assay, as the dead cells turn blue and the live cells will not change color. The cells are examined under a microscope to get a good look of the colonies. As seen on the right, there are various blue cells and many white cells. The blue cells are the dead cells, and the total number of dead versus live cells can be counted and analyzed. However, there was one negative aspect of methylene blue that is highlighted on the next slide. In a comparative study using natural pigments extracted from food and methylene blue in a dye exclusion test on paramecium, it showed that paramecium exposed to methylene blue were all killed within 1 hour, while the natural pigments had no effect. They found that 2 dyes worked as good indicators for viability with the dye exclusion test. These were the monascus pigment, which comes from the yeast rice pictured above, and anthocyanin pigment with tris, which comes from red cabbage. These pigments are a less invasive option for the dye exclusion test than methylene blue, as they do not affect cell health. For an assay that is trying to determine the cell health after exposure to antioxidants, it is not helpful if the dye used in the assay has an effect on the cells itself. In conclusion, for the antioxidant capacity assay, I would choose the TAC assay to perform in lab because it provides a built-in comparison with Trolox to any chosen supplement and allows you to test various dilutions to see which is most effective. For the cell cytotoxicity assay, I would choose the dye exclusion test using natural pigments because I think it is an effective way to determine the health of the cell population after exposure to the supplement, and it is also a contribution to green science with the use of natural dyes. For future research, I would carry out these assays in the lab with yerba mate samples. I would also perform a cell rescue assay with the yerba mate samples, in which I would expose a population of cells to a free radical and see how well my supplement can rescue the cells. I would like to thank the CREATE Symposium for allowing me to present my research today, and give a big thank you to Dr. CY, Dr. HK, and Dr. Simmons for all of the teaching and support throughout this semester. And here are my references. Thank you.